Have you ever wondered what allows you to lift a cup of coffee or walk up a flight of stairs? These actions are the result of intricate molecular machinery in your muscles. Without these tiny components, our lives would come to a standstill. Let's explore muscle contraction and relaxation guided by these molecules. Every movement is orchestrated by a symphony of proteins. Actin, myosin, troponin, tropomyosin, and titin each play a crucial role. Their interaction is a beautiful dance that allows us to move and thrive. Understanding their functions transforms your perspective on your body. Actin is one of the most abundant proteins in our muscles. It forms thin filaments that are essential for muscle contraction. Think of actin as the track on which other proteins move. Without these tracks, muscle movement would be impossible. Actin filaments are like a network of highways crisscrossing within muscle cells. These highways provide the path along which myosin, the motor protein, travels. The structure of actin is like a double helix, similar to DNA but in a different context. This structure allows it to be both strong and flexible, perfect for its role in muscle dynamics. Actin is not just a passive structure, it actively participates in muscle contraction. When a muscle is stimulated to contract, actin filaments slide alongside myosin filaments. This sliding mechanism shortens the muscle, producing movement. The energy for this process comes from ATP, the cellular fuel. Understanding actin's role helps us appreciate how even the smallest components of our muscles are crucial. Without actin, the muscles would lack the structural framework needed for movement. Actin's interaction with myosin is a cornerstone of muscle contraction. Myosin is the powerhouse behind muscle contraction. It converts chemical energy into mechanical force. Myosin moves along actin, pulling muscle fibers together. Myosin binds to actin filaments. The myosin head uses ATP to pull on actin. This action generates muscle contraction. Thousands of myosin molecules work together for movement. Troponin plays a critical role in regulating muscle contraction. It is a complex of 3 proteins that sits on the actin filament. Troponin acts as a switch that turns muscle contraction on and off. Without troponin, the precise control of muscle contraction would be impossible. Troponin has 3 parts: troponin C, troponin 1, and troponin T. Troponin C binds calcium ions, troponin Y inhibits actin-myosin interaction, and troponin T binds the troponin complex to tropomyosin. When calcium ions bind to troponin C, it causes a change in the troponin complex, allowing muscle contraction to proceed. The binding of calcium to troponin C is like turning a key in a lock. It unlocks the ability of myosin to bind to actin, enabling muscle contraction. Without this regulatory mechanism, muscles would either be constantly contracted or unable to contract at all. Troponin ensures that muscle contraction happens only when needed. By understanding troponin's role, we can appreciate the delicate balance required for muscle function. Troponin's ability to regulate contraction is essential for everything from lifting a weight to maintaining posture. Section 5. Tropomyosin, the gatekeeper. Tropomyosin is another crucial protein in muscle contraction. It acts as a gatekeeper, controlling access to the binding sites on actin filaments. Tropomyosin is a long, thin molecule that wraps around actin filaments, covering the binding sites for myosin. In a relaxed muscle, tropomyosin blocks the binding sites on actin, preventing myosin from attaching. When calcium ions bind to troponin, it causes tropomyosin to move away from these binding sites. This movement exposes the sites, allowing myosin to bind to actin and initiate contraction. Tropomyosin's role is to ensure that muscle contraction occurs only when it is supposed to. By blocking or exposing the binding sites on actin, tropomyosin regulates the interaction between actin and myosin. This regulation is critical for controlled and coordinated muscle activity. The precise control provided by tropomyosin is essential for muscle function. Without this regulation, muscles would be unable to contract in a smooth and coordinated manner. Tropomyosin's role as a gatekeeper is vital for the proper functioning of our muscles. The dance of contraction, how actin and myosin interact. Muscle contraction is a dance between actin and myosin. The interaction between actin and myosin is the heart of muscle contraction. Calcium ions bind to troponin, exposing actin binding sites. Myosin heads pull on actin filaments, sliding them past one another. This sliding shortens the muscle fiber, generating contraction. ATP hydrolysis powers the movement of myosin heads. This cycle produces the force needed for movement. Section 7. The role of calcium, the spark of movement. Calcium ions play a crucial role in muscle contraction. They act as the spark that ignites the process, triggering the interaction between actin and myosin. Without calcium, muscle contraction would not occur. When a nerve signal reaches a muscle, it triggers the release of calcium ions from the sarcoplasmic reticulum, a specialized structure within muscle cells. These calcium ions flood into the muscle cell, binding to troponin and causing a conformational change in the troponin-tropomyosin complex. This change exposes the binding sites on actin, allowing myosin to attach and initiate contraction. The presence of calcium is essential for this process. When calcium levels drop, the binding sites on actin are covered again, and muscle contraction ceases. The role of calcium is like that of a key in an ignition. It starts the engine of muscle contraction, enabling the complex interactions between actin and myosin. Understanding the importance of calcium helps us appreciate the fine-tuned regulation of muscle function. Section 8. Titin, the giant protein. Titin is one of the largest proteins in the human body and plays a vital role in muscle elasticity and stability. It acts like a molecular spring, providing the necessary tension and stability to muscle fibers. Without titin, muscles would lack the resilience needed for repeated contractions. Titin spans the length of the sarcomere, the basic unit of muscle contraction. It connects the Z-disc, a structural component, to the M-line, the center of the sarcomere. This positioning allows titin to maintain the structural integrity of the muscle fiber, even during stretching and contraction. The elasticity of titin allows muscles to return to their resting length after contraction. This ability to stretch and recoil is crucial for muscle function. Titin's spring-like properties help absorb the impact and provide stability, preventing muscle damage. Titin's role is to ensure that muscles remain strong and elastic. By providing structural support and elasticity, titin allows muscles to perform repeated contractions without losing their integrity. This protein is essential for the resilience and stability of our muscles. Section 9. Muscle relaxation, the return to rest. Muscle relaxation is as important as contraction. It allows muscles to return to their resting state, ready for the next movement. The process of relaxation is carefully regulated and involves the removal of calcium ions from the muscle cell. When a muscle contraction ends, calcium ions are pumped back into the sarcoplasmic reticulum. This decrease in calcium levels causes the troponin-tropomyosin complex to revert to its original state, covering the binding sites on actin and preventing myosin attachment. The removal of calcium and the covering of binding sites on actin result in the relaxation of the muscle. The myosin heads detach from actin, and the muscle fiber returns to its resting length. This process ensures that muscles are ready for the next contraction. Muscle relaxation is a vital part of the contraction-relaxation cycle. It ensures that muscles do not remain in a contracted state and are able to perform repeated movements. Understanding this process highlights the importance of regulation in muscle function. Section 10. Everyday implications, from gym to daily activities. Understanding the molecular mechanisms of muscle contraction and relaxation has practical implications for everyday health and fitness. Knowledge of how these processes work can inform exercise routines, rehabilitation, and overall muscle health. In the gym, knowing how muscles contract can help in designing effective workout routines. Exercises that target specific muscle groups can be optimized by understanding the roles of actin, myosin, and the regulatory proteins. This knowledge can lead to more efficient and safer workouts. In rehabilitation, understanding muscle physiology can aid in recovery from injuries. Techniques that promote proper muscle function and relaxation can help in restoring mobility and strength. Knowledge of titin's role in muscle elasticity can inform stretching and flexibility exercises. In daily activities, appreciating the complexity of muscle function can encourage better care of our bodies. Proper nutrition, hydration, and rest are essential for maintaining muscle health. Understanding the molecular basis of muscle function can lead to more informed choices in our daily lives. The symphony of muscle function. The intricate dance of actin, myosin, troponin, tropomyosin, and titin allows us to move, live, and thrive. From lifting weights to walking, their coordinated efforts are essential for our daily lives. Understanding these proteins helps us appreciate our muscles' complexity and efficiency. This knowledge informs our health and fitness choices. Think about the tiny molecular machines making every movement possible. Embrace muscle physiology to enhance your health and fitness journey.